recombinant DNA technology topics. Our next session is on screening strategies. In the previous session, we have already discussed cloning strategies. That is cDNA library creation and genomic DNA library creation. Both were discussed in the previous session. Now, after the screening, after the successful cloning of a recombinant DNA, we need some screening strategies for the proper identification of the recombinant DNA construct and subsequent insertion in the vector. Okay. So, here, when we come to the products of the cloning experiment or the construction of the recombinant DNA, which is the product of the recombinant construct, or the recombinant DNA product is the product of the DNA cloning experiment. We have three different possibilities or possible outcomes out of that experiment. The first one is the production of a non-transformed cells. That means the host cells were not at all incorporated with the concerned DNA insert or the vector. So that means majority of the population, for example, in case of a host cell of E. coli, or a transformation of E. coli with a plasmid vector, which is incorporated with a DNA construct, there most of the population of the E. coli or the host cell will not be incorporated with concerned plasmid vector. Only a few of them may be incorporated or inserted with that recombinant construct, which means out of millions of cells in the culture medium, only a few thousand cells may be actually transformed by the experimental DNA construct. The remaining were untransformed or non-transformed cells, and they constitute the majority of the population. Now there are transformed cells containing the unaltered vector. So another possibility is that actually we are cloning, for example, insulin gene in PBR322 and subsequently transfer it into an E. coli host. So the first case is the untransformed E. coli cells without the plasmid vector and the DNA construct. The second possibility is transformed cells with the plasmid. For example, E. coli uptakes the plasmid DNA, that is PBR322, but actually that PBR322 is not incorporated with the concerned DNA insert, for example, insulin gene. In that case, we have a transformed cell with unaltered vector or just PBR322 without the concerned target gene. So that's the second possibility. Now the third possibility is actually transformed cells with the concerned DNA or RDNA. So that is the actual transformed cell with the DNA insert or DNA construct. So out of these three possibilities, or the population in a mixture of or a pool of cells which contains these three categories of the host cells, we have to identify the transformed cells with the actual RDNA construct and eliminate the untransformed ones or non-transformed ones and the transformed ones without the DNA construct. That two populations needs to be excluded from the actual cell mass or that culture medium. So that constitutes the screening strategy. So we require various strategies for the selection of the clones with the concerned RDNA construct. Now, in the case of a selection of the recombinant clones, we can have a selection of clones with the RDNAs or selection of the clones with a specific DNA insert. That means there may be some population of the cells with some unwanted DNA constructs inserted, or some maybe there is some RDNA inserted in that clones, but it is not the RDNA insert of our interest. And there may be some other clones which may be actually inserted with your specific DNA of interest, for example, insulin gene. If you are cloning insulin gene, then you have to identify only the clones with the concerned gene construct. Or here, for example, a specific DNA insert that may be an insulin gene, you have to select that particular clone. So there should be methods for selecting the clones with the RDNA and from which your desired DNA needs to, the clones with your desired DNA insert needs to be identified. And for that purpose, selection of these clones with RDNAs, we have the first step, selection of clones with RDNAs. There we are ensuring that some of the that clones or some population of that cells were actually carrying some RDNA or some foreign DNA. But that first step doesn't ensure that it is your desired DNA. Only in the second step, or the specific DNA construct is verified only in the second step. The first step is the verification of the clones with the RDNA. The presence of an RDNA or a foreign DNA or insert or a foreign DNA construct which is inserted doesn't mean that that DNA construct which is incorporated is your desired one. There may be unwanted DNA fragments or undesired fragments incorporated along with that desired DNA. So you need to eliminate that unwanted or undesired DNA fragment inserts. That clones containing that type of inserts needs to be eliminated. So in this step of the selection process, we need to identify the clones with the RDNAs, and also we have to from among that RDNA constructs, you need to identify the clones with the specific DNA insert of your interest. Here, for the selection of the RDNAs, actually this is not specific for any particular gene type, only it screens for the RDNA constructs. And that the technologies that is used were based on basically reported genes or marker genes. And another category is elimination of the non-transformed cells, then identification of the clones having the RDNAs. So we have various steps and strategies which can be used for selection of such a RDNA containing clones. The first one that is discussed is about reported genes or marker genes. There are, maybe you might have heard about selectable markers and scorable markers. The selectable markers were basically antibiotic resistance genes. For example, kanamycin, tetramycin, ampicillin, or these are standard genes that were actually incorporated into the vector as a selectable marker. So the basic concept is that, for example, in PBR322, we have a tetracycline and ampicillin resistance gene incorporated that one. And you can select the clones that is containing this particular gene carrying plasmid. That may be that only the clones that carry the plasmid with this particular antibiotic resistance genes will grow in a medium containing that specific antibiotic. So by growing in some selective media or some media incorporated with some specific antibiotics will be used for selectively scoring or selectively growing such colonies that carries the antibiotic resistance genes, and the vectors were actually incorporated with some combination of these ampicillin or tetracycline or kanamycin, such type of antibiotic resistance genes. And we will see example later. Anyway, the basis of the antibiotic resistance based selection is the cells that carry the plasmids with antibiotic resistance will survive in the antibiotic containing medium, based on which you can select the genes that is so the colonies that is actually incorporated with that particular plasmid carrying antibiotic. Now, the second category is a scorable marker. Scorable marker means that they produce visible phenotypes. For example, we have a GUS gene that is producing beta galacturonidase gene, which actually based on the blue color production by the colonies. So that is one gene that is actually regarded as a scorable marker. And then another one is a Lux gene, which is also which actually produces a luciferase enzyme, and that based on that luciferase we have a phosphorescence or fluorescence of light emission by the concerned organism that carries this Lux gene. So that is another visible phenotype that is used as a scorable marker. And there is a OCS or NOS, that is octopine or nalopine synthase, which is actually used in nitrogen fixation process. So that genes were also regarded as scorable markers, which results in the production of nodules and which is used for nitrogen fixation. So this type of selectable and scorable markers, both categories were used for initial screening of the RDNAs incorporated in the host cells. So when we discuss about the scorable markers, for example, take the case of the first one, Gus gene, that is coding for beta-galactoronidase enzyme. Here, this is a chromogenic substance, a standardly used chromogenic substance that is used for such selection. And here we have XGAL. XGAL is actually a substitute for such beta-galactosidase production or beta-galactosidase based gene induction. So XGAL can be cleaved by beta-galactosidase, and that colorless compound of XGAL, which is also known as 5-bromo 4-chloro 3-indoyl beta-D galactopyranoside, that is the XGAL. So this compound, which is actually a colorless compound, is actually cleaved by this beta-galactosidase enzyme, and it produces an indoyl derivative. So this process of cleavage by beta-galactosidase of this compound produces an indoyl derivative, which will be subsequently oxidized, and this will be oxidized to a dibromo-dichloro indigo compound, and this compound is actually a colored compound, that is a blue colored compound, which will impart a blue color to the colony in which this process occurs. So this is the basis of beta-galactosidase induction by the XGAL, that is actually a chromogenic substances that is substance that is used for such a selection or selectable or selection based screening of RDNAs. So then there is a elimination of non-transformed cells. So that is another step. For example, PBR322, it is having tetracycline and ampicillin resistance genes for the selection. And then there are another category. So for elimination of the non-transformed cells, one possibility is an antibiotic based selectable marker. And another type of selectable marker or selection process is that phages or cosmids or phagemids or vectors, they are actually packed in virions or phage heads, and that will be used for infecting the host cells. So a host cell, once infected with such a recombinant DNA, recombinant vector like a phage or cosmid or phasemid, actually they are packed in virion heads, so that it's an infective particle. So once it infects the cells, it lyses the cells, and the transformed cells or infected cells will form plaques in the plates. So formation of plaques, colony plaques, that confirms the successful transformation of that host cells by, or successful infection of that host cells by such vectors. So there is another type of another method that can be used for elimination of non-transformed cells. Is it clear? Hello?
Yes, sir.
Okay. So for the identification of the clones with RDNAs, we have a vector with two selectable markers. One that is used for a selection-based screening. And one example I mentioned is PBR322, which contains an ampicillin resistance and a tetracycline resistance gene. And you can place your desired gene. For example, you are cloning insulin, then the insulin gene will be placed in within one of the selectable markers. For example, you will place the insulin gene within ampicillin resistance gene which is present in the PBR322. So that result in interruption or disabling of that particular gene. So once the DNA insert is placed within the selectable marker, for example, ampicillin resistance gene, then that cell will not no more carry the ability to grow in that particular ampicillin containing medium. So a selection process can be arranged by incorporating or inserting the gene of interest in the selectable marker, within the selectable marker. And by the combination of tetracycline and ampicillin resistance, by growing alternatively or by using a replica plating method, you can grow the same colonies in ampicillin containing medium and tetracycline containing medium, and even in both containing medium, so that you can eliminate the cells without any, for example, the untransformed cells will not have any antibiotic resistance gene. So the initial category or the E. coli cells only, that first category cells will be easily eliminated because they cannot grow in the concerned antibiotic containing medium. And the cells which contain PBR322 only, not the DNA insert, they can grow both in ampicillin containing medium and tetracycline containing medium. Whereas if you are placing the DNA construct within ampicillin resistance gene, thereby interrupting it, then the DNA construct containing actually transformed RDNA or that vector containing cells cannot grow in ampicillin containing medium because that gene is interrupted. Then that results in the difference in the expression of that particular two types of antibiotic resistance. So that means PBR322 containing cells will that can grow in both antibiotic containing mediums, whereas the RDNA containing constructs can only grow in tetracycline containing medium, not within ampicillin containing medium. So that means we have there are a selection based mechanism now available for screening out your RDNA constructs, and this is about the selectable markers, especially PBR322, which is based on two selectable markers or two antibiotic resistance genes, which is placed simultaneously within the vector. And another category of identification is based on complementation. Complementation is actually it's a actually the vector contains a part of the gene actually that is missing in the concerned host cell. For example, I will describe it in the next slide with an illustration. So complementation, in complementation, the vector which is carrying the DNA, RDNA, actually that vector actually contains a part of the host cell gene which is actually missing in the host cell. Okay, the host cell in this case actually lack except alpha gene. So the alpha component, the alpha portion or a portion of the LAC-Z gene is missing in the original host cell. So the original host cell cannot express LAC-Z gene. So that needs to be complemented by the vector. Once the vector carries this LAC-Z gene along with its sequence, then the host cell will have the complete sequence available for the production of the LAC-Z, and so then that expresses that particular LAC-Z protein. Here, the insert is actually placed within this LAC-Z gene. So just like that of the gene that is placed within the scorable markers, if you are placing an insert within the LAC-Z gene in the vector, it interrupts that gene. So the vector which is carrying the RDNA will have a non-functional LAC-Z gene, so it cannot complement the vector, complement the host cell. So if the vector only LAC-Z alpha gene carrying the vector only enters the host cell, then it can complement the production of alpha, lag Z alpha. Whereas if that lag Z alpha is interrupted by the new DNA insert, then it will no longer be able to complement the production of lag Z synthesis. So in this case, what happens is that vectorally clones will be blue and successful transformants will be white. Here we have the illustration that is actually it is based on insertional inactivation. Here you can see that the lag Z gene in the host cell, there is a lag Z alpha portion is missing. So we have a host cell, specifically designed host cell, in which the lag Z gene is missing a portion. So lag Z needs to have two components. So the first component is actually produced by the host cell, whereas the second component was the host cell cannot produce this one because it is a deficient mutant. Whereas your vector actually it carries that part of the lag Z. So lag Z alpha part gene is carried by the vector. So if the vector is incorporated into the host cell, then the missing portion of the lag Z alpha, that alpha peptide is produced, so that the functional beta galactosidase is produced, and that functional beta galactosidase can effectively cleave the X-gal, and the colonies will turn blue. Okay. And in the second case, where we are placing the insert, for example, the insulin gene that you want to clone, that is actually inserted within the lag Z alpha of the vector. Thereby, this alpha component is also blocked. So if a successful transformant or the transformant with the concerned DNA insert within the lag Z gene is constructed, then it cannot produce the lag Z alpha, and that means there is a non-functional beta galactosidase. Thereby, colorless colonies or plaques will be produced. So that means this is the basis of the blue-white screening in the cloning. So the blue colonies were untransformed ones with the it may be transformed, but the transformed cell actually it doesn't contain the RDNA. If the RDNA construct is inserted successfully, then it will turn colorless or white. So blue-white screening is a standard procedure in which the blue colonies were the colonies that contains vectorally sequences and not the RDNA. If the RDNA construct is actually inserted within the vector, then we will have white colonies which produces white color colonies or plaques. So this is about the insertional inactivation that is based on lag Z gene, and here the color is actually produced by the conversion of the X-gal by the beta galactosidase, and the fully functional beta galactosidase is produced only when there is a complementation that is done by the lag Z alpha carried by the vector. So if your insert blocks that sequence, then thereby alpha peptide is not produced in the transformed cell. So that means a successful recombinant with the RDNA will not have the functional beta galactosidase gene. So or beta galactosidase protein is not produced, and that type of colonies or the plaques will turn white, which can be easily identified in the screening process. Is it clear?
Yes, sir.
Yes, sir.
Okay. Now, identification of the clones with the RDNAs. Again, there are methods for direct selection. So here you can insert codes for a gene product which is defective in the auxotrophic host. So that is another method. So you can insert codes for a gene product which is not actually present in the host. Normally you know that auxotrophic cells or auxotrophic host were actually defective in production of something, at least in production of some amino acids or some deficiency will be there in the auxotrophic host. which needs to be supplemented in the growth medium so that the cells will grow. So such type of auxotrophic host can be incorporated with some specific gene production codes. For example, if you want to screen some, say, some cells, say, insulin containing, insulin is actually incorporated into an E. coli cell. So you can have an auxotropic E. coli cell created, and it will be deficient in, for example, production of a particular enzyme or something. Anything can be missing. Any vital component of the host cell can be missing, and your vector may be incorporated with that particular gene, which will supplement the auxotrophic host. So once the gene is inserted along with the vector, then that will supplement the auxotrophic host. Thereby, it doesn't require that supplementation in the medium. Usually, host cells are grown in deficient medium. Only transformed cells can grow in that deficient medium. Okay, so say a particular amino acid or any containing amino acid is deficient in the medium, and that deficient medium cannot normally support the auxotrophic host. So if a gene for the synthesis of that particular amino acid is incorporated into the system through the vector, then only the transformed cells which is carrying that genes which is required for the production of that deficient product, for example an antibiotic, an amino acid as explained, and that cell will grow in the deficient medium. So the auxotrophic host, they are normally supposed to grow in a medium supplemented with that particular component. It cannot grow in a deficient medium because the cells cannot synthesize that one. So in that case, if you are incorporating the gene for the production of that deficient component in the vector, and subsequently the vector is actually incorporated and transformed into the and that gene is transferred into the auxotrophic host, then there will be a complementation of that gene, and that concerned deficient product will be synthesized by the concerned transformed cell. So thereby it can grow in the deficient medium. So this is how some direct selection method can be incorporated in the selection process. Another one is a suppression of nonsense mutations. That is another method, and I am not going to discuss further. So another one is insert a gene in lambda vector lysis suppressor gene. So that is another example in which, for example, lambda GT10, that's a lambda vector, and now this lambda vector is having a gene for lysis repression. So normal lambda GT10, once it infects a host cell, it will not lyse the cells because there is a lysis repressor gene which is present in lambda GT10. Okay, because of the presence of a lysis repressor gene, the normal vector cannot lyse the cells. A successful transformant will rise to clear plaques because the insert is placed within the lysis repressor gene, thereby disrupting the function of the lysis repressor protein, which results in the lysis of the transformed cells. So successful transformants will give to clear plaques only, and others will give cloudy or turbid plaques because there is a lysis repressor, functional lysis repressor in the normal vector. But once it is incorporated with the DNA construct, within that lysis repressor gene, that protein will be disabled. So if without the lysis repressor, the cell will be lysed by the concerned phage. So this is another example of direct selection. Then there are lambda replacement vectors and cosmers, which lack the minimum required size for packaging in virions. Usually, you know that these phage heads were packed by the concerned DNA during the packing of the virions, and the packing of the virions require a minimum size. For example, maybe 25 kb or 30 kb. That size is required based on the type of the vector. Some minimum required size is there. A few thousand base pair like the sequence cannot be directly incorporated or packed into a host into a phage head. So the lambda replacement vectors actually they lack the minimum required size for packing. So this vector only that cannot be packed into the phage heads. But if the vector is incorporated with the concerned DNA, target DNA or RDNA, then its size increases. So the vector lambda replacement vector maybe it is only 10 kb, and the minimum required size for packing on virions maybe that is 30 kb. So if you are placing a 20 kb insert within the lambda replacement vector, 10 kb vector, then it constitutes the total required size, that is a 30 kb, then it can be packed. Then only it forms the, it can be applied for a successful transformation or subsequent cloning process. So this is how a lambda replacement vector can be successfully used for cloning of your target DNA, that's or desired DNA. So lambda replacement vector only will have a minimum size which cannot be directly packed. So if the vector is incorporated with the concerned RDNA, its size increases, which reaches and thereby it reaches the minimum required size for the packing or phage head packing, and that will be packed and that can be used for transforming the host cells. So this is another method of a direct selection. Now when we come to the selection of a clone containing a specific DNA insert, the previous ones were basically selection of the clone or elimination of the untransformed ones or selection of the transformed ones with the RDNA. But our intention is to select the clones containing specific DNA insert, not any insert. So there comes the importance of sequence dependent screening, and you also require screening of expressed protein products, because our target is cloning of a specific gene with a specific product. We are not looking for clones with clones which is incorporated with any DNA. We are looking for clones with a specific DNA. There we have different sequence dependent screening techniques. One is colony hybridization, and then nucleic acid hybridization, probes, then chromosome whacking methods, screening by PCR, then differential screening techniques, gene tagging. So these are all different sequence dependent methods or DNA sequence based screening methods. And we can see them one by one. The colony hybridization, as you know, here we have the original transformed colony is growing on an agar plate. So from this one, you can create a replica plate onto a nitrocellulose disc. Replica plate, that plate into a nitrocellulose disc. Then that disc is removed. Then it will be lysed with NaOH and neutralized, the protease is suppressed, washed, so that the final result is that DNA fingerprint of that particular colony which is attached on this nitrocellulose membrane will be created after this treatment. So then you can hybridize it with a P32 labeled probe, which is specifically designed for a DNA based P32 labeled radioactive probe, which will target a specific sequence. So DNA hybridization is actually sequence specific. So if you are looking for a particular gene, for example insulin gene, then you can design a probe against the insulin gene and target with P32, then try to apply on this nitrocellulose membrane. So only the colonies or the DNA extracted from the colonies will bind to the concerned probe, which is labeled with the concerned radioactive label. So a specific sequence specific interaction is occurring here, and that will bind here. So an autoradiography will give you the spot which contains the colony, successful colony, which is carrying that particular sequence. And you can retain the master plate here, and from looking to the master plate, you can locate the colony and pick this colony for as a positive colony for a subsequent cloning process. So this is about the colony hybridization. Is it clear? Hello?
Yes, sir.
Okay. Another method is chromosome walking, and a chromosome walking is a technique that produces sets of overlapping DNA clones for studying segments of larger DNA that can be cloned individually. So, usually such larger DNA sequences cannot be cloned in a single step because there is a size limitation for every vector. So in that case, such larger DNA or longer lengths of the DNA sequences can be studied by constructing overlapping fragments or the clones containing overlapping fragments, so that individually they can be arranged and sequenced or analyzed. So such type of chromosome walking technique is it's a set of procedure that is it uses it produces overlapping DNA clones, and that can be used for studying larger fragments of the DNA. A method for it's a method for analyzing of a larger regions of the DNA in which each end of a single large single cloned DNA fragment is used separately to screen recombinant genome library for other clones containing neighboring sequences. For example, there will be a sequence, and you will take the end of one sequence, and using that sequence you will try to find other sequences which will have some overlapping region, or the same region containing clones will be identified, and if such a colony is identified, from that point other regions can be located. You can see that here the initial step is usually restriction digestion of the entire region of the DNA of interest. So you can digest it with multiple enzymes like OR1, SAL1, or in combination of different enzymes also you can create different combination of lysis products of the genome, which results in all different types of fragments. So mixer lysing of different enzymes will create a large number of different size fragments in the mixture. So there is every possibility that there is a overlapping fragments. So for example, one fragment may be created from the first base to 500 base pair, and another fragment may be 100 to 1000, and another may be 500 to 2000. So there are a lot of fragments which is created with different lengths, and there is a very possibility that such fragments will be overlapping. So the overlap can be identified only if you identify one of this end of one sequence, identify the end sequence of one sequence, and look for such similar sequences in the other regions. So in other fragments, if there is an overlapping regions, you can place it on the top of that one, so it will be extended. And in such manner, you can, for example, here you see that here is the larger chromosome segment. This is a desired destination region. So this is a starting location. So you identify one clone with a starting location, and you take the sequence at one end. The fragment first region, the first segment will be identified, and you take this sequence and you construct a primer and you identify this sequence, and based on this sequence you can identify other products which contains this sequence. So there are other, for example, three different products in the mixture. One starts from here and ends here, and another starts from here and ends here. And you can see that the third fragment is actually the longest one, or it is the farthest one. So starting from your desired location of the first end, this fragment gives you a higher length. So you can pick this one as the possible candidate and select the second. Start the second step with this second end. So the second will be used for identifying the third segment, and again it is probed, and this segment is identified, and the third segment end will be identified, and again this process will continue. So by step process, you can identify the clones with overlapping fragments so that you can construct the entire length of the chromosome. Or the desired length of the sequence can be identified. So this process is known as chromosome walking. So by a combination or a mixed digestion of restriction enzymes, you can construct fragments of different lengths, and you can identify the overlapping regions in each fragments and arrange them in order, so that you see at the final stage you can have different lengths products arranged, and they can be probed with the different probe. The first probe is based on the first end, So this is the second probe, this is the third probe, fourth probe. So each probe will be constructed based on the previous sequence entry, and the products will be identified and they can be arranged in a sequential manner. So this is the concept of chromosome walking. So the next technique is based on PCR. It requires sequence information, as you know. PCR requires specific primer, and specific primer requirement actually it demands knowledge of the sequence. Without the knowledge of the sequence, target sequence, you cannot construct a primer and you cannot go for PCR. So it requires sequence information. And usually you can transfer the clones or individual colonies into multi-well plates or microtiter plates, and each well can be treated with lysis enzymes, and the DNA can be individually screened. So each well can be screened by PCR. So once a successful target well that contains the colonies is identified, that well can be or the cells from that sample can be diluted further and in multiple wells, and again that can be grown and again conduct the PCR, so that the successful colonies containing the recombinant construct can be identified using such PCR. So in this case we will use multi-well plates, for example 96 well plates, and grow the individual cells or the samples in each plate, and by PCR you will screen for the concerned target sequence in each and every plate or each and every well, so that once you identify a well with a positive sample, you can take the sample and further dilute it and again split into another 96 well plate. So the sample dilution will distribute the cells uniformly, and finally you will get a pure colony containing the concerned DNA construct, and that can be identified by PCR based method. So that's the concept of screening by PCR. Now there are differential screening based methods in which to identify cDNAs that are differentially expressed in two cells or cell types or at different developmental stages, etc. You can use this method. So differential screening is required if you compare the gene expression. Example, isolate gene expression in response to salt stress. So your target is isolate gene expression in response to salt stress. So you will prepare two sets of replica filters and probe the first filter with the mRNA or cDNA isolated from the cells in normal condition, which is a control. And then you will probe the cDNAs, mRNAs isolated from the cells that is grown in salt stress condition. So there is a developmental stage difference or cell type difference or differential expression. Anything can be done. So developmental stage or cell type, for example, pancreatic cells or blood cells, two types of cells, and how the gene expression differs, that can be identified. Or the growth conditions like normal growth condition and salt stress condition. So it will have two different sets of cDNAs or mRNAs expressed. So that can be probed. So two sets of replica filters will be created, one with the normal and another with the desired altered condition. So that can be compared. The results of the colonies can be compared by differential screening method. And one of the extended version of this differential screening can be just like microarray screening, etc. There are other advanced methods are there. So the basic concept is that you can identify differentially expressed cells or different cell types or expression in different cell types or expression of genes in different developmental stages on or various environmental conditions, growth conditions, etc. Everything can be compared by taking two different sets of samples from different cell types or cells grown in different conditions. And that type of screening is a differential screening. So this is a plus minus method in which screened with the probe or screened with the minus probe, plus probe or minus probe. So this is one strategy. So different colonies will be screened in such a in large numbers, and this is a part of the differential screening method. And another one is gene tagging. to isolate those genes that produce detectable phenotypes, but whose base sequences or protein products are not known. So that's the purpose of gene tagging. We want to isolate genes that produce a detectable phenotype. For example, a flower color or antibiotic production or anything, any particular gene type or differential detectable phenotype. And you want to isolate that particular genes, and sequence it. But for specific identification and sequencing, you don't know the sequence of the gene. You know the phenotype. The expressible detectable phenotype is identified, but the gene is not located. In that case, you can tag the gene. How it can be done? Use integration of the DNA sequence or tag, or a non-sequence or a tag sequence can be incorporated into the clones. So the clones actually that is attached with the tag and the detectable phenotype. So that clones can be easily identified. So your first step is the attachment of a tag sequence, non-tag sequence along with the vector in the cloning step. So during the cloning step, you will attach a tag sequence or a non-DNA sequence, and then you will attach the different products of the detectable phenotypes. So out of the many clones that is created, maybe some of them may be your target DNA sequence. That is the gene of interest of your detectable phenotype. So that will be attached. You can identify the clones based on their expression. So such clones with your tag sequence attached with the detectable phenotype can now be isolated, and they can be probed based on the tag sequence. So if the tag sequence is there along with the detectable phenotype, then you know that this sequence is actually tag sequence is attached with the phenotype region. Now they can be combined with inverse PCR. That's another method. I think we already discussed that in the PCR variants. So inverse PCR can be used for sequencing and analyzing these unknown sequence. So this is an example of the inverse PCR, in which say a known sequence is attached as a tag to the unknown sequence. So for example, by incorporating this one, now this detectable phenotype is now silenced. Okay. So disabling of this detectable phenotype and successful incorporation and PCR or a screening based PCR screening based identification of this non-sequence ensures that the desired gene is silenced by the insert, and the insert is now present here. This construct can be circularized, then it can be lysed from the center, and on both ends now we have the non-sequence. So once it is linearized, on both ends we have the non-sequence, and that non-sequence can be used as the or used to amplify based on the primer is designed against that sequence. So your non-sequence attached there were actually used for constructing the primers to amplify, and this can be amplified, cloned, and even sequenced. This sequence can be determined using this method. So this is one of the methods that is based on gene tagging. And another one is nucleic acid labeling. Nucleic acid labeling is, as you know, this image shows a nucleotide triphosphate. This is a phosphate, sugar, and base. This base region is available for pairing with A. Now, this is a spacer region, and this is a deoxygenin, that's a labeling compound. So this label is attached to the nucleotide triphosphate. So this type of nucleic acid labeling is available. This type of labeled nucleotides can be used during the amplification step. So this is a primer, and the labeled nucleotides is provided in the medium, so that in the reaction mixture. So the sequences synthesized here will be incorporated with the labeled nucleotides, which can be identified. So the labeled nucleotides, maybe that's a colored compound, and that can be detected based on the the quantity of the amplification product. So the labeled nucleotides will be incorporated during the amplification stage. That's one of the methods. And another method of nucleic acid labeling is radioactive labeling, where the 5' end is attached with a phosphate, radioactive phosphate, which will be attached at the end, and that can be again used for identification and autoradiography based identification of the sequences. So this is a nucleic acid labeling, either dye-based method like this one, or that is actually based on PCR-based amplification and subsequent incorporation of that labeled nucleotides in the amplification products, or you can attach labeled P32 labeled ATP that is attached at one end, and that phosphate label at the 5' end is acting as the label for the identification of the strands. So this is about nucleic acid labeling. And nucleic acid hybridization reaction is also there, where we have single-stranded DNA probes against specific genes. So we construct a specific DNA probe against your specific gene, and at specific condition, hybridization conditions, you will have, for example, hybridization in 50% formamide and 42 degrees Celsius. You will have a specific, this reaction condition varies because based on the reaction conditions and the sequence, length of the sequence and various other factors, the parameters will change. So at a particular stage, we will have the specific products attached and non-specific binding will not be promoted. Whereas, for example, in this case, if you bring down the temperature from 42 to 35 degrees Celsius, there will be imperfect binding. And so the result of the hybridization reaction actually depends on the reaction conditions and other factors. So this is another step in which this can be used for identifying the introne sequence, regions of the intrones in a eukaryotic DNA. So this technique is used, for example, this is the mRNA of this gene. So you denature the DNA and hybridize it with the mRNA. So this RNA, if once it is hybridized, the introne regions actually they will form a loop because it cannot pair with the RNA. So this RNA DNA hybrid, the things that is a single stranded or the region that is present as a single stranded region, that will be the introne regions. That can be cleaved by the S1 nuclease treated because S1 nuclease, as you know, it removes the single stranded DNA regions. So that, again, that can be constructed. And if you go for a gel electrophoresis, you will have the exons separated here. The untreated DNA control is here, which is having higher weight. So this one, and the exon 1 and 2 will be represented here with lighter weights because it is cleaved. The S1 nuclease will remove this region of the introne and the exons will be identified. So exons can be easily identified and introne region can be removed and using this method. So this is another method of nucleic acid hybridization available. So there are various methods of gel transfer based techniques, etc. And another one is expression based screening. In expression based screening, we have complementation, unique gene products based and antibodies specific to protein products, then colony or plaque screening with antibiotics, antibodies, then fluorescent activator cell sorter based methods, southern or western screening, and then northwestern screening, other approaches. So there are various expression based screening methods are also available for selection. For example, here we have one antibody based method in which the protein product that is expressed will be tagged, will be specifically located by the antibody against that protein, and again a labeled protein against that antibody can be tagged there, and autoradiography can be used for detecting that colonies which carries this antibody tag. So such type of autoradiographs can be constructed and specifically identify the protein. So it's a protein. The product-based screening is there. It's not sequence-based, but specific protein antibody interaction is used for identifying that colony.